Okay, last up for the workshop, we're going to have an hour-long talk by Natalia Komarova from the mathematics department at UC San Diego. So Natalia got her Ph.D. in applied math at the University of Arizona and held positions subsequently at the IAS, Rutgers, UC Irvine, and moved to UCSD a few years ago. So she's going to talk to us about mathematical modeling in spatially fragmented populations. So.
Thank you so much for inviting me here. I really enjoyed my stay. Beautiful place and great company. So today's talk is not really game theory, but it has elements that may be interesting for some of you. So we're going to look at birth and death processes, and many of them will feature frequency-dependent fitness. And yesterday we discussed that that's an important part of game theory. We'll have multiple types, wild types and mutants. We'll look at competition between different types. The focus is evolution. And in this talk, I will focus on comparing well-mixed and spatially distributed or fragmented population. So this is a well-mixed population that is evolving. Okay, and most of evolutionary theory has been done for this because it gives rise to beautiful mathematics, and you can actually solve a lot of it. As opposed to this, we have spatial populations where individuals know who their neighbors are and they interact with their nearest neighbors or next nearest neighbors, their neighborhoods. And the proxy to this, and a nice way to study some of the properties of spatial populations, is fragmented populations. So we have islands where on each island individuals interact like well-mixed, and there's a degree of communication among the islands through migrations. So we'll look at all of these. And my biggest claim here is that a lot of properties of evolution completely change when we move from well-mixed to spatial populations. So that is kind of the main idea of this talk. And so this slide here illustrates, these are real images. It illustrates that, of course, everything is spatial that we're looking at. There are all sorts of examples here. The bacteria, this is a aerial view of Everglades. So these are trees, this is lichens. This is some species of fish, invasive fish somewhere. And I don't know what that is. I took this picture. So first, I'll tell you how I got into this. And this is a very long story. So this is my introduction. I'll talk about mostly my very recent work, but this is old work and what got me interested in this whole topic. So fitness valley crossing. In cancer, a lot of cancers start by a sequence of two events. Here is a cell with two copies of intact gene, and then the first copy gets inactivated. But this cell is not a fit cell. It's the opposite. Its fitness goes down. But when the second copy of the gene gets inactivated, this is a supercell. It has a very high fitness. So this is what we call a fitness valley crossing. To go from here to here, you go through a stage of low fitness, and that takes a longer evolutionary time. So this is fitness landscape. If you turn it upside down, and if you're a physicist, this becomes a potential barrier that you have to overcome. And you go from here to here. So we studied this process a lot. And there are kind of a couple of different things that can happen here. You start from a population of cells that all are in this state. And then one of the purple mutants appears, and maybe it spreads. It kind of takes over. And after a while, one of them creates a red mutant, and that spreads. But that doesn't happen very often. More often than not, before the fixation of the purple types, you get the second mutation, and you get into the red mutants. This is what we call stochastic tunneling, and we looked at the speed of that process. So this is an approximation that we used, which I'll use later in my recent work, which is coarse grain approximation. You go from everybody green to everybody purple to everybody red. And you can calculate the rates of transition among these states. So the first process that we considered is a non-spatial Moran process. So a disclaimer, I don't really like Moran process anymore, and because it's finicky. In this process, everything depends on whether it's birth-death or death-birth or other microscopic assumptions of this. But this is a very nice process where you can do a lot of things analytically. So the population is constant, one dies and another one reproduces, and you just keep going like this. And you stop when you get the first mutant of the red cell. We calculated the rate of that. Let's take a note. This is a proxy of the speed of evolution, how quickly you create the super mutant, right? And now let's go to a spatial model. And this is 1D space. Again, a Moran process, where now, if somebody dies, this spot can be only replaced from one of the neighbors. So it goes like this. And you stop when one of the super mutants appears. So we calculate the rate of this happening in space. And this is faster. Why is it faster? You see, U2, U is the mutation rate. And you have this very small, it's a probability. And we have this probability to the power 1/3 for space. And in the well-mixed process, it was this thing to the power 1/2. So this happens faster, and you can prove it, at least in this very, very simple setting. So this is kind of a general result. You can look at 2D and do the same thing in the Moran process. Okay, so I'll show you some numerical results here, depending on whether you only reproduce into your neighborhood or maybe several layers. The time to achieve this double hit mutant grows. So the rate is the fastest when you're the most spatially restricted. So what do we conclude from this? We conclude that having these spatial interactions accelerates evolution. And there's a very good intuitive reason for this. I'll go over this quickly, but look here, we compare a mass action system where single mutants kind of pop up anywhere, regardless of space, because we don't even consider this in the simulation. And here we reproduce into nearest neighbors. So if you have a mutant, they tend to exist in clusters. So if you're a weak intermediate mutant, you will agree with me that it's much better to be in this situation than here, because here you are competing with everybody in the population to be the next person who replaces an empty space. And here you are competing with mostly people that are as weak as you are, so you don't feel so bad. So weak mutants that exist in islands like this have a longer existence time. They stick around for longer, they feel better, and they will produce the super mutant faster. So this is why space accelerates evolution. So that was a beautiful result. And we kind of summarized all these results. The arrows indicate different. So this is no space, 1D, 2D, 3D. These results are due to Duret. So we have this beautiful correspondence between the speed of evolution and dimensionality. The fastest happens in 1D. This one. No, where is it? Here. This is the fastest. If you increase dimensionality, it goes a bit slower, and the slowest is mass action. So I love this result. It reminded me of PDEs. Any of you who like PDEs, you know that Green's function has a completely different functional form depending on the spatial dimensionality. So I thought, oh, great, this looks fundamental. So that's what made me interested in this topic. So this first old result is that evolution is accelerated because of the existence of spatial interactions. So today I want to talk about 3 case studies that are all very recent and some unpublished, that go through various scenarios and show what else happens in space compared to what happens in well-mixed populations. The first story will be about scaling laws for neutral and advantageous mutants. That's an advantageous mutant. The second story is about accumulation of disadvantageous mutants. And the third story is about quasi-neutral mutants. And I'll tell you what that is. Scaling laws for neutral and advantageous mutants. So these pictures are real and they are experimental. And amazing, they look better than almost any computer simulation that we have. These are colonies of various organisms, bacteria or yeast, that grow out. So they increase in their radius and they mutate. The mutants are shown in color. And we kind of watch it, and we want to know how many mutants we expect to have in a colony of a given size. So these are simulations, and it's amazing, right, that simulations look worse than the real life. So there are various situations that one can consider. Let me tell you what the spatial process is. This is not a Moran process. So cells are placed on a grid. Each cell has a division and death rate. So wild types are W and DW for division death rate, and mutants are M and DM for mutants. We have a continuous time birth and death process with mutations that go one way. Upon division, a cell chooses uniformly any neighborhood cell. Like, for example, if it's nearest neighbors, it's going to be the 8 nearest cells. Pick one at random, and if that's occupied, you don't do anything. But if it's free, you put your offspring there. And also you have a probability to die yourself. So this is the process. It's a natural, not constant population size process. So we kind of start with one cell in the middle, and we'll let it grow out, and we color mutants yellow, and we watch them grow. So the first publication about this was in Halachek, was by Halachek and group in 2016. They found some very interesting patterns. So the question that they asked is, do we expect to have more or fewer mutants in a spatially growing colony like this, compared to a colony that grows exponentially without any space, just mass action? Here's a result for this neutral mutant that grows. So in a mass action or well-mixed colony, this is how many mutants you observe. And you compare it with a spatial colony, you have a lot more. So somehow, in a colony of the same size, if you grow it in 2D, you're going to have a lot more mutants, even though all the rules are more or less the same in the two. So there's an explanation for this, which is about jackpot events. These are very special mutations that give rise to many, many mutants. So let's suppose that we have an exponentially growing colony. Start with one cell, forget, suppose there's no death. One cell gives rise to two, two give rise to four, and so on. And if it so happens that at the very beginning, one of these cells mutates, all the downstream cells are going to be mutant. It gives rise to a huge mutant clone. And in this case, you're going to have a lot of mutants when you stop your process and count the mutants. But of course, that doesn't happen very often. In this, mutation rate is very low. This requires one of the very early cells to do that. Most of the time, you're going to have something like this, right? Mutants happen at the end. There's only a few of them. So jackpot events are very rare in well-mixed populations. But now if you look at the spatial population, here's the spatial population. You start here, it's a 1D space. That's my favorite space because it's the most drastic. It's the most different. Okay, so you start here, cell divides, this one divides and so on. And so the population grows like this. And at some point, the mutation will happen. And then everybody is going to be mutant, right? So almost every mutation event is a jackpot event because it gives rise to a long colony, like everybody else after you is going to be mutant. So that's pretty much why in a spatially growing colony, you're going to expect a lot more mutants. This is 1D, it's very, very severe, but things like this also happen in 2D, right? And in other spatial scenarios. So basically, we want to see, does the same result hold for advantageous mutants? How about different geometries, different dimensionalities, and multi-hit mutants? So yes, these are neutral mutants look like this. They give rise to slices like this. Advantageous mutants give rise to expanding slices. Disadvantageous mutants give rise to little bubbles like this. So the answer is yes, pretty much the same thing happens for advantageous mutants in increasing populations. You see spatial colony produces more mutants than in non-spatial for advantageous mutants. And we have different scaling laws depending on the dimensionality and geometry. So we have 2D flat, so it's like a rectangle advancing this way. 2D range, this is mostly what I've been showing. 3D flat, a cylinder advancing this way. 3D range, a globe growing in 3D. And depending on whether you're disadvantageous, neutral, or advantageous, you can kind of figure out how many mutants you expect to have in a colony of size n, where U is the mutation rate here, and you have all these different powers. So we did that, but we didn't really understand it. Now we understand it better. Let me show you. It's it's very easy to see how to derive these laws. So in 0D, in the absence of space, things grow like this. X dot equals Rx. And this gives rise to an exponential growth. Okay. How about in 2D? What's the difference? In 2D, X stands for all the cells inside this circle. But the ones inside don't divide. The circle only expands through its surface. The surface scales with square root of X, right? So in the right-hand side here, instead of X, you have square root of X. You solve that, and you get T squared. So spatial colonies in 2D grow as T squared. In 1D, you have this. They grow as T to the 1. In 3D, they grow T to the 3. You solve the ODE. So these are the growth laws of one type of cells in 1D, 2D, and 3D. Okay. Now let's put mutants in. How do you put mutants in? In 1D, let's suppose we have this rectangle, and the cells grow that way. At some point, a mutant happens, and everybody else is going to be a mutant on average, right? So we do have a stochastic process. But on average, every mutant is going to give rise to kind of a mutant population that doesn't expand on average or doesn't decrease because it's a neutral mutant. It has the same properties as the other cells. So we don't expect these things to become thicker or thinner. So we can just simply integrate. If it starts at R and goes to R, this is the size of each of these. And then we integrate them together, and we get the growth law or the scaling law of the number of mutants. It scales with this R squared. Okay. Advantageous mutants are expected to give rise to colonies that are expanding because they're advantageous. So you can perform the same calculation and then get the contribution R cubed. Very simple. And S is the advantage. Now you can do it in 2D, slices like this, or expanding slices like this. And you get a very nice, very kind of simple number of growth laws. So I already explained to you that wild type goes as T squared, or T cubed in 1T, 2D, and 3D. For neutral mutants, you have T squared, T third, T fourth. Depending, so one more power of time. Advantageous mutants add another power of time. So you can show all this by this simple type of calculations. Okay, so now you can look at double mutants, double hit mutants. So here you have one type, and it can give rise to another one. Right? So you can perform similar calculations and figure out scaling laws for any case. And well, we didn't have growing colonies in our lab, but we can grow them in a computer, right? So we can see, okay, we have a computer simulations. Do these laws actually hold in simulations? And you can see that here, like you can measure the power. Oh, by the way, when I say T, I mean time. So it grows as a square of time or cube of time. It's the same as the radius of the colony, because the colony itself grows, the radius grows proportional to time. So if something scales with T squared, it's the same as R squared. So here we expect to have R cubed, and you get R cubed. Here you expect R fourth, and so it works, right? And you can do it for any number of mutations, one hit mutant, two hit mutant. You can get it for any number of advantageous or neutral intermediate steps. So there's a general formula that you can have. So for example, for neutral mutants, depending on 1D, 2D, 3D, and depending on how many steps you have, this is the exponent, right? Mutants go as colony radius to the power of A. This is the power A. You have all these different possibilities. If you have 3 mutational steps, one of which is advantageous, so you can go neutral, neutral, advantageous, or neutral, advantageous, neutral, and so on. So for all of this, you can derive these laws, and they all have very kind of symmetric, nice shape. Why do I even do this, right? So I'll show you why. So you can do all of this by simulation and calculate how many, on average, 1 hit, 2 hit, 3 hit mutants you can have in a colony of any given size. So let's take a look at this picture. We have, this is the number of single mutants in the colony of a given radius, double hit mutants, and total. The thick lines are my simulations, and the lines are theory, my scaling laws. They work pretty well. But you can see that the simulations stop here. And why do they stop? Because when the colony radius reaches, what, 400, the total number of cells is 10 to the 7. And you can't really simulate anymore, especially if you want to get some statistics. You can simulate once, but okay. And my theory is cheap. It can go to any size, right? And we can see what happens in very big colonies for different scenarios, different number of mutants, different mutational pathways. For example, we can start answering some practical questions about cancer. So one of the things that exists in the theory of cancer is the existence of mutators. These are cells that accelerate the mutation rate that gives rise to de novo mutants. So let's suppose that we want to know how many mutants of a given type you have in a tumor. So we start with some type of cells. One mutational event gives rise to a mutant. We want to know how many of these we are going to have in a given size. Another way to achieve this, and that's what they say often happens in cancer, is that first, you acquire a different type of mutations that makes the cell a mutator. So this cell mutates faster. So this cell will produce those red mutants at a higher rate. So you can either go directly and mutate, or you can first become a mutator and then create the target mutant. So is this a viable pathway that can even be observed? Because see, here you have just a single step, and here you have 2 steps. Each of them is relatively rare, right? Mutation rates are very small, as was just mentioned, like they go from 10 to the -7, 10 to the -9, right? So having 2 steps, requiring 2 steps on top of each other is a big deal. So does this even matter? So you can use the theory to see how this pathway weighs compared to this 1-step pathway. How likely are you to see a mutant that appears through a mutator compared to a direct pathway? So this is measured here by this quantity Q. The closer it is to 1, the bigger the contribution or probabilistic contribution of this double-step mechanism. And so this is plotted as a function of the population size. Let's look at the green line. The green line corresponds to exponentially growing tumors. No mass action, well-mixed system. The green line that measures the contribution of these mutators stays around 10 to the -6, no matter what you do. Right? So in a non-spatially well-mixed colony, you will never see a contribution of mutators. Now you go to 2D and 3D, and you can see that depending on the parameters, you can have a pretty robust contribution of that mechanism. And this is only because this is a spatial colony. The laws of mutant accumulation are different. And this is what we can show by this theory, right? Except it's not that simple. When I told you about mutators, if you actually look at real mutators that happen, say, in colon cancer, it doesn't work like this. So 13% of colon cancers are characterized by what they call microsatellite instability. Microsatellites are repeated sequences like this. It's easy to make a mistake when you copy that. So a mistake associated with that is not corrected in cells that are characterized with this microsatellite instability. These cells are denoted by stars here, but to get here, it doesn't just take one mutational event. This is wild type. You have to go 1, 2, and only then are you mutator. And then you have a really fast rate to create the mutant. So it's a higher cost to go from wild type to mutator. So what is faster? To go 1, 2, and mutator and now fast compared to just one step. Let's see. So in the absence of space, exponentially growing colonies, the contribution of that mechanism is 10 to the minus 11 or so compared to 1, right? So never see this. But and you can play with parameters, right? But in 2D or 3D, you start noticing very robust contribution of this mechanism for realistic population sizes. So for cancer, what's a realistic population? Detection level is about 10 to the 10. So after that, so tumor grows to 10 to the 12, 13. So these are realistic population sizes. So yes, in space, you see the contribution of these because of the different scaling laws that happen in space. So for neutral and advantageous mutants, my conclusion is that we have different scaling laws in different dimensionalities. In well-mixed systems, we never see multi-hit mutants. And in space, they become very likely. Okay. Now, disadvantageous mutants. That's my second story. So this is, you can find this new work in bioRxiv, but don't read it because it's where we've just improved it. So the next version is coming up. And so what we study here is disadvantageous mutants. So first I want to motivate it. Why would we even care about disadvantageous mutants? They're not viable, they're deleterious, they don't do anything. So let's suppose that this is your growing type, and it creates these deleterious mutants. This two-dimensional fitness landscape tells you the fitness. So these are deleterious mutants. They have smaller fitness. But then something happens, and one of these mutants can actually become advantageous. Why? Like you start treatment, right? You start treatment of cancer, and now a resistant cell that was kind of disadvantageous when the cancer was growing, now you apply treatment, that's the only one that does grow. So deleterious mutants can become advantageous if circumstances change. So it's important to know what they are. And also they can pave the way to future mutations. As we already saw in Fitness Valley Crossing, it can be like a stepping stone toward another mutation that gives you a lot of fitness. So it's important to know what these guys are doing. So that's the question we ask in this little project. So again, we want to compare a non-spatial accumulation of these mutants and a spatial. So this is actually even simpler than before. We don't even assume frequency-dependent fitness. This is just a linear birth-death process that is shown here. We have one-way mutations in this linear birth-death process. And the spatial process is the same as I already told you. We just use the same rules. We start from one cell, we grow, stop at a given size, and count the number of mutants that we obtain. So like this. And so there is a specific question that we have in mind. So this is my growing colony, and it makes these disadvantageous mutants. Many of them will die, right? So these are the red ones. And we stop at N. How many mutants do we get? And the question I pose is, how does this depend on the death to birth ratio of this colony? So as this thing grows, let's think what happens if you change the death to birth. In the absence of death, it takes a certain number of cell divisions to go from here to here. How many? It's N, right? You start with one cell, no death, you stop at N. I guess it took N minus 1 cell divisions. If you turn on death, so increase death to birth, then the number of divisions to go here will increase. because you have to compensate for death. So here's a simple argument. You increase death to birth, there are more cell divisions, you expect to have more mutants. Okay, let's see. But before I proceed, you're going to ask me, Natalia, what are you doing? This is Luria-Delbruck distribution, at least without space. Studied continuously for more than 80 years, since 1943. One of the foundational mathematical models of evolutionary genetics. Hundreds of dedicated papers. A lot of great people worked on this, right? So there is a lot of literature devoted to this. And I especially want to note a contribution of Tibor Antal, our colleague. This is very useful. However, nobody asked the question I'm asking. So I looked. So there are many models, almost none of them do any theory in a spatial colony, but without space, nobody really cared how this depends on death to birth ratio. But this is an important issue, right? How exactly the colony grows. So let's do this. So the easiest way to study the well-mixed system is to write down ODEs. I love ODEs. They give us intuition. So this is my ODE. Normal cells divided rate R, and they mutate and they die. Mutants appear at rate mu, and they divide and they die. And this rate is like smaller. They are disadvantageous. You can grow this ODE to a certain size, count the number of mutants. You solve this analytically, right? And Tibor Antal proved that as n goes to infinity, and if you ignore jackpot events, this is the correct system to look at. However, there are jackpot events, and I'll show you, even in the absence of space. This is the probability distribution of the number of mutants at size 10 to the 4. So actually this is missing from this because this is a log scale, but there should be another bar that corresponds to 0 mutants. Most cells will have 0, most simulations will have 0 mutants. Some will have 1 mutant, 2 is the case like this. But look at this here. It's 10 to the 4 mutants, so everybody is mutant. Why is that? It's because at the beginning, some cell mutated and replaced everybody. So it does happen. And so we can't ignore this. So our talented postdoc, Samrat Mondal, derived an expression that takes into account exactly this. So we can amend the result from ODEs and get the expected number of mutants in the stochastic process by taking into account the probability of that. So I don't think it's been done before. And it works pretty well. Okay, it's an approximate theory. This is the mean number of mutants at size n in the stochastic process are given by dots. Samrat's theory is green line, and this here is deterministic. So there's a big difference sometimes. Okay, so let's see what happens to the number of mutants as a function of death to birth ratio. So I forgot to mark the horizontal axis. This is death to birth ratio. No death, high death. So you can see that if the mutants are disadvantageous, the number of mutants grows with death to birth ratio. That was our simple intuition. Increase death, it takes more cell divisions to get to size n, you will have more mutants. So the simple intuition holds in this well-mixed system. Actually, only if disadvantage is at birth. I don't want to give you too many details, but if the mutants are disadvantageous in death, if they die more, then it can go the other way around. But now I want to compare this with the spatial system. Now we grow them in space. Again, we grow a colony, stop at the same size, count mutants. How does this change with death to birth ratio? Here's a simulation. So this is the size of the colony at which we stop. And the different lines correspond to different death to birth ratio. So for small sizes, like 10 to the 3, death to birth ratio increases, the number of mutants increases. And then you can see that these lines kind of cross over and go the other way around. For large populations, death increases and the number of mutants decreases. So because we're in space, this simple relationship can completely reverse. So, and this is kind of a summary of what we see in the spatial system. So there are different regimes. Here, we increase this colony size, and we increase disadvantage. So neutral here, strongly disadvantageous here. This can increase with death to birth ratio, decrease with death to birth ratio, or be non-monotonic. So this here is when it decreases. This is kind of this weird regime. It corresponds to strong disadvantage and big population sizes. So the conclusion here is that in well-mixed systems, the number of mutants increases with death to birth ratio, and in space, it can decrease. So again, space gives you something completely different. Why are we studying disadvantageous mutants? So one of the reasons is that they can give rise to evolutionary rescue. So what is that? So cancer grows, these mutants pop up, you start treatment, and if everybody is susceptible, treatment will be successful. But if one of these mutants is resistant and it happens to be there, then it can rescue. Well, rescue in this case is bad. Okay, so what is the probability of that? Actually, it's a completely different problem, because to calculate the expected number of mutants, this is the probability to have I mutants, that your formula, it's important, like the whole tail of the distribution is important. A rescue, you multiply by this is the probability of I mutants to make it. The tail of the distribution is completely unimportant, so it's a slightly different problem. Samrat Mondal, my postdoc, again, derived a beautiful, very simple formula for the probability of evolutionary rescue in a well-mixed system. And you can see that as the death to birth ratio increases, probability of rescue decreases. Why is that? Because if there's a lot of death in the system, these mutants that can be there have a much reduced chance to make it because they die more. So increased death to birth ratio, probability of rescue decreases. Now let's do it in space. And you can see that it can be non-monotonic or increase. So in the spatial colony, in the colony that grows with a lot of turnover, dying and divisions and death, this very important measure starts depending differently on the parameters. So the other conclusion for disadvantageous mutants, in a well-mixed system, probability of rescue decreases with death to birth, and in space, and it can increase. I'm down to my last topic, which is quasi-neutral mutants. I'll tell you what that is. And actually, this goes beyond quasi-neutral mutants. But so let's talk about different types of mutants. We already talked about disadvantageous and advantageous. Here, we can map it out like this. So let's suppose that this is division rate, and this is death rate, and this is what the wild type is. Wild type has this division rate and this death rate. And compared to that, if a mutant has like an increased division rate or a decreased death rate, it's advantageous. If a mutant has an increased death or decreased birth, it's disadvantageous. So on this two-dimensional parameter space, you can map out all the mutants. And most of the time, we kind of go along one of these lines. We either increase or decrease the death rate, increase or decrease the birth rate, and making them advantageous, disadvantageous. But really, you can put any point here and you get something. Right? So there's some evidence that both of these change simultaneously in mutants. So this one shows that mitotic index is correlated with apoptotic index, which means that mutants that die more also divide more. So you can simultaneously increase division rate and death rate or decrease both, right? So this gives rise to this area on this graph. So inside, like if you put a straight line here, these are mutants which with proportionally increased divisions and deaths. Right? They have the same birth to death ratio, but they either go faster or slower. So we call them quasi-neutral mutants. So let me show you something very simple. So again, I love ODEs. Let's take a look at the wild type that divides at this rate and dies at that rate and grows according to logistic growth. And this is a mutant. So what are the equilibria? If birth to death ratio of the wild type is bigger than that of a mutant, you have a competitive exclusion. The one that has a bigger ratio will win. If the inequality is the other way around, the mutant will win. Okay, so this is kind of equivalent to expected number of offspring during lifespan. Okay, now let's suppose that they're the same. And now in the ODE, we have a neutrally stable line of equilibria, where anywhere here is fine. So ODE doesn't really tell you what happens to such mutants. It looks like a degenerate case, but you will see that I will expand this to something that is less exotic. But let's talk about these. So these are quasi-neutral mutants. So we're going to study them stochastically. And this is the process. So this is a nonlinear birth-death process where the divisions happen according to logistic growth. And this is kind of the space where we're working. So we have the number of wild types and the number of mutants, not a Moran process. So these two are independent. So it's a two-dimensional Markov chain, which is kind of limited by this carrying capacity. If the mutants are disadvantageous, you're more likely to go here and they go extinct. Advantageous mutants kind of shift here. And we want to know what happens to quasi-neutral ones. Right? So if you run simulations, so this is again the same space. Each point corresponds to a certain number of wild types and mutants. And let's suppose that tau, the acceleration factor, is 100. So mutants divide and die 100 times faster. So it's like the motion in the vertical direction goes really fast, and the motion in the horizontal direction is slow. So you're more likely to kind of die here. And so both of these axes are absorbing. Once you touch it, you're done. Okay, so this makes mutant extinction a little bit higher. If your mutant is 100 times slower, you go very fast in this direction, and you're more likely to find your death here, which is mutants taking over. So Josh Plotkin published a paper with this group. Very beautiful calculation, the diffusion approximation that tells you the probability of this happening. So what is the probability of mutant fixation in this case? So N is the population size, mean population size, and tau is the acceleration factor. And the beautiful formula tells you the probability of mutant fixation. We also looked at that in the Moran process. We didn't know about it, actually. It kind of happened by chance. Amazingly, it's exactly, so this here, exactly the same formula, but only for death birth. If you do birth death, and depending exactly how you arrange it, you can get anything, more or less. So I don't like Moran process. But nonetheless, so we have this formula. What does it mean? So in the real neutral mutants, so one of the very well-known facts of genetics, the probability of mutant fixation, if the mutant is neutral, is equal to its initial frequency. So if you start with 1 out of n cells, the probability of neutral mutant fixation is given by 1 over n. Okay, so I think many people will know this. You can prove it from symmetry considerations. Everybody out of n cells is the same. Who is going to be the primordial Eve? Anybody is equally likely. So 1 over n is everybody's chance. So quasi-neutral mutants have a multiplier on top of this. And so if tau is the acceleration factor, if tau is greater than 1, this factor is less than 1. So for those who are accelerated, the probability of mutant fixation is less than neutral. And for slow quasi-neutral mutants, the probability of fixation is greater than neutral. But are they really advantageous or disadvantageous? Let's check. So let's plot the probability of quasi-neutral mutant fixation as a function of the population size. So this here, the orange line, I'm plotting 1 over n as a function of n on a log scale. So it looks like this. This is 1 over n. Neutral mutants do this. Quasi-neutral mutants that are accelerated take that and multiply it by a constant number. So the blue line looks like this. And this here, green, is truly disadvantageous. So you can use this formula, plot this as a function of n as for different, this is the mutant advantage R. So for disadvantageous mutant, this dependency becomes strictly exponential, right? So on a log scale, it looks like a decaying exponent. So my quasi-neutral mutant is not disadvantageous. It's just a scaled neutral. And let's look at slow mutants. So again, this is 1 over n. Slow quasi-neutral mutants have a factor that is greater than 1. So this is the blue line, it's higher. But truly advantageous mutants have a horizontal asymptote. So if you're truly advantageous, your probability of fixation becomes constant as a function of population size. So they don't do that. And so they remain quasi-neutral. So the point of this talk, of course, is to see what happens when we add spatial interactions. So here we'd first add a proxy of spatial interactions, which is fragmented population. We are going to split my big population into a bunch of islands and allow for slow migration between them. And we're going to see what happens first to these quasi-neutral mutants. And this is what happens. So I'm plotting the probability of mutant fixation as a function of the population size. So if the population is non-fragmented, you have this line, we've already seen it. It's 1 over n on the log scale, scaled with a constant factor. But now when my population is split, you have this. My mutant is fast, it's 10 times faster, and it behaves completely like a disadvantageous mutant. And look at the difference, right? This is the same mutant in a well-connected population, non-split population of the same size. And this is the mutant that is exactly the same, but it lives on islands. Look at the difference here. This is 10 to the minus 5, this is 10 to the minus 3, right? Two orders of magnitude. You change parameters, it's huge difference, right? So it starts behaving completely like a disadvantageous mutant, just because we fragmented the population. And this guy here is a slow quasi-neutral mutant. As a function of population size, its probability to fixate becomes constant. So it acts completely like an advantageous mutant. And again, look at this, 10 to the minus 4 difference between this is non-fragmented and this is fragmented. So fast quasi-neutral mutants behave like very disadvantageous if you put them on islands and allow slow migration. And slow quasi-neutral mutants behave like advantageous mutants in these populations. So we have a nice approximation for this. This is actually, we have analytical results for this one. Because we're looking at a fragmented population. It's in real space is harder, but for fragmented population, we came up with this coarse-grained approximation. So these are my islands, and they're wild types green, mutants purple. There is slow. There's that. So this is similar to your talk. So in each island, the dynamics is kind of fast, and then when migrations happen, we pretty much assume that things are settled within each deme. So that's this is a slow migration approximation. They're weakly connected. So because of this approximation, we can say, okay, this is completely green, and this is going to be completely purple, right? So each deme only has one type, and then there's slow migration in between. So this trick, so this is a very highly dimensional stochastic process because you have to keep track of the number of mutants and wild types independently in each island. So if you have D islands, you have D times 2 dimensions in your stochastic process. Not so great. This trick turns it into a 1-dimensional Moran process, but your object is the number of fully mutant islands. So we only have one variable. We have to count how many purple islands we have. And this lives in 1D, and there are transitions in between. And we derive a very kind of concise formula for this. This is the acceleration factor. This is the probability of mutant fixation in a single isolated gene. This is probability of a wild-type fixation in a mutant gene. And this is what we call a relative fitness of a mutant gene. So this goes into the Moran process formula. That gives me probability of fragmentation. Works like magic. This blue thing is a theory, and the dots are simulations. So this is really nice. And I want to advertise a little bit. I now collaborate with a very talented postdoc, Yifu, and with Ruth Williams, my colleague, who is a famous probabilist at the University of California, San Diego. We are collaborating on this. They found that you can describe this by using methods that come from stochastic chemical networks. And so they're doing this now, but completely rigorously. So we're going to have all the proofs within a month. So yes, this is biology giving rise to mathematics, I think. Yeah. So, but the formula is correct, right? So we derived it. Now we're proving it. Let's take a look what it gives us. So we have one island of a certain population size. We can split it in different number of sub-islands with migrations. Depending on this, how much you split, things happen. So probability of fixation. So start from the dashed line. As the gene size decreases, so we go from here to here. If your mutants are fast, the probability of fixation decreases. Whoop. And if your mutants are slow, it increases. And you can measure gene fitness in this way. So gene fitness decreases or increases for slow mutants as you increase the degree of fragmentation. But of course, nobody is quasi-neutral, because that whole machinery kind of required that the mutant division rate scales as tau with the wild type division rate, and death rate the same tau. Let's relax that assumption and make mutants truly advantageous or truly disadvantageous. So let's suppose for now that tau is equal to 1. So this is just your normal way to assume that your mutants divide 1 plus s times slower or faster than normal cells. And now we multiply by tau, which can be acceleration or deceleration. So this is more or less kind of, this is a two-dimensional parameterization of this. So that can describe any point here, right? So, but theory actually works for all of this, right? Which is very nice. And this is what we have. So let's suppose that S here is a selection factor. So we heard about this. So here I take 10 to the minus 2 selective advantage. So mutants are actually advantageous. We multiply the division rate by 1 plus S. They are 1% advantageous. Big difference in evolutionary biology. And so their probability of fixation is denoted by this point is higher than neutral. But now we're going to turn on this factor, tau. We're going to make them a little bit faster. They still remain advantageous. The expected number of offspring they produce during a lifespan is still 1% bigger, but they're now faster. And look at this. The probability of fixation goes down, 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 and they actually become disadvantageous hugely in the fragmented population. So this is all only works so by turning on this turnover factor, making them accelerated in space, in the fragmented population, we turn the force of selection, with the sign of selection changes. These advantageous mutants become disadvantageous. And the same thing happens if you, so here the mutant turnover factor goes from 1 to 0. So these are slowing down. They are originally disadvantageous, negative S. So they have their probability of fixation is less than neutral, but we make them slower and slower and slower and slower. They become advantageous. So in the presence of space, we see a reversal of selection. So this is this. theory works very well with stochastic simulations performed in the islands. We can now do a truly spatial model. The same as I described before. We have a grid, divisions, deaths. Let's see what happens here. No theory, but the same result. So here we have a disadvantage of 0.1% and no acceleration in a spatial population. And this mutant behaves like it's disadvantageous. It's selection mutation balance. It's there at small numbers, it gets produced, but it gets outcompeted. And now let's turn on some deceleration. So now the same disadvantageous mutants does everything 10 times slower. And look what it does. This mutant goes up and up and up and outcompetes. It becomes advantageous. Complete reversal of selection in space. Here we start with an advantageous mutant, which outcompetes the wild type like it should, but then we'll make it faster, and it doesn't anymore. It behaves like completely disadvantageous mutants. So here are my conclusions of the whole talk. So I hope I convinced you that spatial interactions, including fragmentation, can fundamentally change evolution dynamics. So first, they make evolution faster. That's the first result. Fitness valley crossing goes faster. For neutral advantageous mutants, mutant accumulation obeys different scaling laws in space. The lower dimension, the more likely we are to see multi-hit mutants. For disadvantageous mutants, space reverses the dependence on death-to-birth ratio. And finally, in this last part, I showed you that space can even reverse the force of selection, making advantageous mutants into disadvantageous and the other way around. So with that, I want to thank my group, especially the two postdocs, Yifu and Samrat Mondal at UCSD, Ruth Williams and Dominic Waters are my collaborators at UCSD. Thank you very much.
Thanks, Natalia. Questions?
For the fragmented populations, I didn't quite understand what you meant by fragmented because you never really told us the migration rate. I guess it's less than 1/n or something like that.
So the migration rate is assumed to be so slow that the dynamics inside each gene will most likely come to its resolution before the next migration event occurs. So in the absence of de novo mutations, let's put a mixture of wild type and mutant cells into a gene. They will sort it out one way or the other before the next migration event occurs.
Yeah,
so
that's Mn less than 1, something like that. Yeah, I understand.
Yeah,
thanks.
So something like this will hold if you crank up the migration rate, but the theory becomes like the approximation gets worse. Yeah.
Can I ask something on that? Similar question about that model is you didn't mention the spatial topology between the islands. Is the migration local in 2D or is it uniform in space?
That's a great question. So very interestingly, it doesn't make a difference. Doesn't make a qualitative difference. So we tried migration in mass action ways, so anybody can migrate to anybody else. We tried the circle, so absolutely no qualitative difference. And right now, the new theoretical paper that is coming up, we'll do it on any graph. So we set a graph of mu ij is the rate of migration from island I to island J. Yeah, it all. Yeah.
Really, that surprises me because I was thinking, there's a continuum limit of an infinite number of islands, but you can take that limit in many ways, and you're saying it doesn't matter.
So okay, so when you start talking about the number of islands going to infinity, now we have to be careful. Because an important assumption is the smallness of the migration rate. So if everybody is migrating from an infinite number of islands, that may break. So you have to be careful there. But the assumption of what I just talked about, the time scale of things that are happening in a single island should be faster than time scale of migration.
I'll let someone else ask. Do I have more?
Yes. Thanks. That was very interesting. One big difference for me is kind of that in the Moran process, normally I don't think of a growing population. And everything you do here is kind of for a case where I think of a growing population.
So not in the islands. So in the islands, you could do a Moran process because each island has a carrying capacity. So each island has a carrying capacity, but we don't keep them coupled. So it's a naturally demographically fluctuating rate. So this last problem, we first did it with Moran and got crazy results depending on exactly what you assume about migration. So when you migrate in a Moran process, you can just give birth into another island, or you can swap and you can change birth death to death birth. Some of these results don't hold.
Yeah, I mean, I think there are more things known, like the circulation theorem, for example, this of Ares-Lieberman, that transfers to these island models. So that shows that in some structures you will see an effect, but in some not. But I think what is more interesting is actually you really focus on growth, which might be the biologically more relevant case than the
island. So
but in the rest of
the other ones, it's a growing population. Yes.
Yeah, and I think that's something that is for me very different from the Moran process where you have these weird results. Okay, thanks.
Hi. Really nice results. I was just curious if you could like clarify tau that I think you call like acceleration factor. I'm just trying to kind of grasp what it would mean in context. Like, is it just like a higher birth death ratio, or like what does a FASTA mutant?
So we have I and J, then go to I plus 1, J, for example. And that time T with rate I are wild type. So this is wild type increasing, right? In the linear process, say. And now J goes to J plus 1, J are mutant. So these are the rates, right? So we are saying that, and similar with death. So we have RM. They are proportionally scaled. The mutant division rate is, tau, is tau times bigger than the wild type division rate. The mutant death rate is tau times bigger than the mutant death rate. So they are proportionally scaled. And tau can be greater or smaller than 1, so mutants are either accelerated or decelerated. But this is not correct because it's actually this.
Another question. Thanks. I guess in the first part of your talk, you mentioned how those images were so amazing. You know, they're better. They look better than simulations. But did you try to extract data from them, or has someone else tried to do that? Can you try to compare it to your predictions? Because I imagine you can try to extract from that something like maybe you can extract the dimension of the system, or you can extract the significance of the mutations. Like, I don't know what's already known kind of a priori.
Halacek and his group started this whole thing, and they grew these. So they looked at neutral mutants in two-dimensional range expansion. So that case was done experimentally, and it works, right? So it matches his theory, it matches our theory. We expanded the theory to different types of mutations, different geometries, and different numbers of dimensions. For the other ones, we only have simulations. It would be nice to look, yes, but
I mean, I guess a little bit behind my question is it truly 2D? It looks smooth there, but like if you zoom in, is the edge rough? Because the 2D assumption is that you have this perfect circle, right?
The rough edge is kind of the most difficult part of this problem, which I completely ignore. And a lot of theory has been attempted. I don't think there are too many conclusive results. And the rough edge is something that I'm very interested in.
Well, I was thinking maybe you can infer the dimension if it's a fractal dimension, because your theory seems like it would extend naturally to that case.
Yes, we're trying to figure it out.
Great. Anyway, very nice work. Thanks.
Thank you.
Could you say a little more about how the stochastic chemical reaction network theory can help in providing sort of rigorous results here. I guess I have a little bit of intuition of what sort of the deterministic theory does and like the zero deficiency theorem and stuff. And that's not sort of giving me a picture. So I was just curious if you can expand on that.
So in the stochastic chemical reaction setting, they usually don't think about evolution or populations or islands or anything like this. They're reactions, and you assume that there is a small parameter. And what you find is you find a system in a long-lived quasi-steady state. So it lives there for a very long time. And then at some point, there is a drastic event, and then a transition that happens, and the system finds itself in a different state. So this corresponds. So when I first saw this, like these people were, this is what they do, right? So when I first saw this, I thought, okay, this is my islands that can turn color, right? So these are long-lived states. So in the limit, when the migration rate is 0, everything equilibrates, and some islands will be mutant, and some of them will be wild type. And now let's put a little bit of migration rate, and then a mutant can come from this island and go to a wild type island, and maybe turn it in a different color. And then again, we wait for a long time. So the system state changed, and it lives there for a long time until the next migration happens. So these chemical reactions that they always talk about, here, their role is played by migration and population. So I think it's a very beautiful connection that has not, I don't think it has been done before, but as far as I know, yeah.
I do have questions.
Oh, thank you.
I think Josh was.
No, it's okay.
I want to follow up with Jeremy's question. So the first thing that come to mind when you talk about stochastic chemical reaction network was, I was thinking, in your model, would you take into account the complex biochemical reaction network, within the cell, and sort of show robustness? Okay, because one of the main question there is to show sort of robust result that does not depend on the reaction rate, but only the topology of the reaction network. So I would say there's.
This is a good point. Nothing much depends on the migration rate because you take a limit of that going to 0. So you have this disjoint absorbing states when it is equal to 0, and then they turn it up a bit, and then you get the first correction. So you know how the probability of fixation scales with that epsilon small parameter. You know how the time to fixation scales with that. But it's not chemical reactions. It's an analogy.
Okay. But very interesting. Thank you.
I think Josh had a question.
Yeah, I was wondering something towards the very end, Natalia. You showed a simulation with like a Moore neighborhood, like a real simulation. This is for the third part, quasi-neutral mutations in space. But you also showed you have analysis for dividing up the plane into different numbers of Deans in the low migration limit, and you get different results for different numbers of Deans. You showed us that. But presumably, like in some limit of the number of Deans getting large, you'll get closer to like real continuous space, right? So is there some sense in which that's happening? Or
so this limit is a little difficult. So first of all, you cannot take the island size to 0.
To 0, yeah.
Because what are you going to do when the sizes, when the carrying capacity is 1? So things break down a little bit. So somehow you decrease the Dean size in the fragmented model as much as you can. And then you take your continuous agent-based model and you can increase the neighborhood size. You can take this neighborhood and I don't think there is an exact match simply because these neighborhoods are not isolated with each other. And for sure, there is no such thing as weak connection between the Deans. We were able to do the theory with Deans because of the small migration rate. In the real spatial simulation, they are very well connected, but the qualitative result holds. And I think that's the lesson. I don't think we can achieve quantitative correspondence, but we have a qualitatively the same result. Yeah. like always. It's not the first time like we encounter this. So the same phenomenon is observed, but of course we can't really get the qualitative part.
It's just a follow-up on that question. It seems like it would be an interesting and not too difficult homework exercise to do like the death-birth process on a cycle with, you know, one cell per location with a quasi-stationary or quasi-neutral mutant. And just, you know, maybe that would be another route to getting analytical results, or have you tried that already?
I tried this in the first project with crossing Fitness Valley. That's how I got into this whole spatial thing. But so let's see. It's not very easy, I don't think, but I think maybe you can do this. Okay, why don't you do this? No, yeah, sound interesting. Yes, yeah, thank you.
Any other questions? Okay, thanks. Oh, sorry, one more. I'll bring it over to
My question depends on the local nutrient concentrations because in your model you fix the growing rate somehow and the mutation rate, but it depends on the local constraint, mechanical constraint, and the nutrient constraint. And for example, if the growing rate increases, it means that it needs more food, but there is another threshold for the diffusion of the food to reach that point. So somehow I cannot understand that part because the increasing or growing rate cannot be constant or differentiation and other rates.
Yes, you're absolutely right. So a tumor can only go up to 2 millimeters inside before it needs angiogenesis to provide nutrients. So it summons blood vessels from the vicinity to provide its own nutrients. Otherwise, it develops a necrotic core, and so everything inside is dead. So organoid tumors can only be very small because they don't have that luxury. Real tumors can avoid that by doing what I just said, angiogenesis. You're right, real tumors are more complex, and yes.
Okay, now we can thank her again.